When we talk about hormones, it often feels like we're just um, scratching the surface of something incredibly vast, really complex. Yet these tiny chemical messengers, they're kind of the silent architects of so much within us, our development, our daily experience, even, fundamental aspects of our identity. And perhaps few are as central or as frequently misunderstood, let's be honest, as testosterone. So today we're embarking on a true deep dive into the profound world of testosterone and its fellow androgens. We're going to venture far beyond the simple headlines of popular perceptions to genuinely grasp what makes this hormone tick, how it meticulously shapes us throughout our entire lives, and the intricate ways it's now harnessed and understood in modern medicine. Our mission, truly, is to arm you with the kind of comprehensive, nuanced understanding. Well, the kind that makes you feel like an elite professional in this field, ready to truly master this vital information. We want you to walk away from this deep dive with a deep, confident grasp of the science.
And yeah, to achieve that level of mastery, we're pulling from really the gold standard. GOODMAN and GILMAN's The Pharmacological Basis of Therapeutics. This isn't just a casual overview. We're going to systematically explore every crucial detail, from the molecular pathways that, govern these hormones right down to their far-reaching clinical applications. We'll trace their origins, their incredible physiological impacts, the significant consequences when they're absent, and the sophisticated ways we intervene with therapeutic preparations and targeted antiandrogens, too. We'll even delve into the intricate pharmacology behind treating conditions like erectile dysfunction, really ensuring no stone is left unturned in our quest for a truly complete picture.
That sounds like an exhaustive and incredibly valuable journey. Seriously.
Yeah.
So let's begin at the very beginning then. Before we even talk about the immense impact of testosterone, we need to understand its genesis. Where does this incredibly powerful hormone actually originate in the body? What are its primary sources?
Yeah, it's a great starting point because understanding the source helps us appreciate its journey and influence.
Hmm.
Testosterone, which is the principal androgen secreted by the body, predominantly originates in these specialized cells known as Leydig cells.
Leydig cells, okay.
Yep, these are found within the testes in men. And these Leydig cells, they're incredibly efficient biochemical factories. They synthesize the vast majority of testosterone through a series of very specific and intricate enzymatic pathways. It's a precisely orchestrated process, really. Starts with cholesterol, moves through several precursor molecules.
Right, yeah.
Now, while men undeniably produce significantly more testosterone, it's crucial to remember that testosterone is also the principal androgen in women.
Okay, but
Very. In the female body, it's synthesized in two primary locations. Within the corpus luteum, that's a temporary endocrine structure that forms in the ovary after ovulation. And also in the adrenal cortex, which is the outer part of the adrenal glands, perched right above the kidneys. Both of these sites utilize similar, though distinct, synthesis pathways to produce testosterone, albeit in much, much lower concentrations compared to men.
Got it. Lower, but still the main one for women, too.
Exactly. And it's not just direct synthesis we need to consider. There are also important precursors. Molecules like androstenedione and dihydrotestosterone, often just called DHEA. They're weaker androgens in their own right, but what's fascinating about them is their versatility. They can be peripherally converted into testosterone in various tissues throughout the body.
Oh, interesting. Like a backup supply.
Sort of, yeah. Acting as a kind of reserve or supplemental source for the hormone. This peripheral conversion mechanism, it adds another layer of complexity to how the body manages its androgen levels.
So testosterone isn't just a fixed presence. Its levels fluctuate wildly across a person's life, from before birth right through to old age. This sounds like an amazing story in itself. Can you walk us through that incredible lifespan journey and how those levels shift?
It truly is a dynamic story, and this variability is absolutely key to understanding its effects. One of the most striking observations is the profound difference in testosterone secretion between men and women across nearly all life stages. And this directly accounts for many of the distinct physiological characteristics we recognize as sex differences.
Makes sense.
So let's start right at the beginning, in utero during fetal development. Around the 8th week of gestation, the fetal tests in males, stimulated by placental human chorionic gonadotropin, or HCG, they begin to secrete testosterone. This initial surge is incredibly important. By the start of the second trimester, say around week 13 or 14, these fetal testosterone levels are remarkably high, actually approaching concentrations seen in mid-puberty around 250 nanograms per deciliter.
Oh, that high, that early.
Yeah, this early and robust presence of testosterone is absolutely critical. It's the driving force behind the initial male sexual differentiation. It ensures the development of male-specific internal and external genitalia.
Right, setting the stage.
Exactly. Now, as gestation continues, these levels do fall by the end of the second trimester. But interestingly, they rise again to about 250 ng-del right around birth. This second fetal peak is believed to be influenced by luteinizing hormone, or LH, secreted by the fetal pituitary gland, stimulating those Leydig cells once more.
Okay, another surge.
Then, after birth, there's a brief dip in testosterone, followed by another distinct peak, again around 250 ng-del, at about 2 to 3 months of age. People sometimes call this the mini-puberty of infancy. Its consequences, well, scientists are still actively trying to fully understand them.
Fascinating.
Following this, testosterone levels typically drop to below 50 ng-del and remain relatively low until the onset of puberty. Puberty itself, which generally kicks off in males around 12 years of age and continues until about 17, is marked by a truly dramatic increase in serum testosterone concentration.
The big one.
That's the one. As men move into early adulthood, their levels stabilize, ranging typically from 300 to 800 ng-del, and this is a stark contrast to the much lower and relatively steady 30 to 50 ng-del seen in women throughout their lives. This enormous magnitude of difference in male testosterone during puberty is, of course, responsible for the profound and visible pubertal changes that further differentiate males. Everything from muscle development to voice changes.
Right, all those things we associate with puberty.
Precisely. And the story doesn't end there. As men age into what we call senescence, their serum testosterone concentrations gradually decrease. It isn't a sudden drop, more the slow, steady decline. This age-related decrease in testosterone is increasingly recognized as a contributing factor to various other effects of aging, things like changes in body composition, energy levels, even bone health, which we'll definitely explore in more detail later.
That's a truly profound shift across the lifespan. Really highlights just how dynamic these hormone levels are. So once testosterone is secreted, how exactly are these levels so precisely regulated, and how does it actually travel around the body? Because I understand it's not just floating around freely, there's a sophisticated transportation system at play, right?
You've hit on a critical point. The body has an incredibly intricate feedback system to regulate testosterone. It's really quite elegant. In men, the principal stimulus for testosterone secretion comes from luteinizing hormone, or LH.
LH, okay.
LH is secreted by specialized cells in the pituitary gland called gonadotroph cells. Interestingly, follicle-stimulating hormone, or FSH, which also comes from these gonadotrophs, might potentiate or sort of enhance LH's effect on testosterone production. Now, the secretion of LH itself is under the command of the hypothalamus, a region in the brain. The hypothalamus releases gonadotropin-releasing hormone, or GnRH.
So it's like a chain reaction. Hypothalamus, pituitary, tests.
Exactly. A chain of command. Hypothalamus to pituitary. Pituitary to testes. And here's where the elegant self-regulation comes in. Testosterone itself directly inhibits the secretion of LH from the pituitary.
Ah, the feedback loop.
That's the one. A crucial negative feedback loop. It's a finely tuned mechanism that ensures the body maintains testosterone levels within a relatively narrow and healthy range. If testosterone gets too high, LH drops, which reduces testosterone production. If it gets too low, LH increases, stimulating more testosterone. Simple in concept, complex in execution.
Makes sense. Keeps things balanced.
Precisely. And this regulation isn't just about static levels, it's quite dynamic. LH is actually secreted in pulses, occurring roughly every 2 hours. These pulses are notably stronger in the morning, a rhythm that was clearly observed in studies, way back.
Oh, interesting. A daily rhythm.
Definitely. And this pulsatility appears to stem from the equally pulsatile secretion of GnRH from the hypothalamus. In a mirroring fashion, testosterone secretion also follows this pulsatile pattern and exhibits a diurnal rhythm, meaning it changes over a 24-hour cycle. Plasma concentrations are typically highest around 8 a.m. and lowest around 8 p.m.
So morning is peak time.
Typically, yes. However, these characteristic morning peaks do tend to diminish as men age, which contributes to that overall decline we discussed earlier.
Right, the age-related changes.
Now, once testosterone is secreted and makes its way into the bloodstream, it doesn't just travel on its own, it's largely chaperoned by specific proteins.
Ah, the transport system.
Exactly. Approximately 38% of circulating testosterone binds with high affinity to a protein called sex hormone binding globulin, or SHBG.
SHBG, got it.
When it's bound to SHBG, the hormone is considered biologically unavailable. It's like it's in a locked vault, unable to interact with cells.
Okay, so it's tied up.
Pretty much. Another significant portion, nearly 60%, binds with a lower affinity to albumin, which is a more general transport protein in the blood. This leaves only about 2% of the total testosterone unbound, or free.
Just 2%.
Just 2%. And it's this small free fraction that is generally considered biologically active, meaning it's available to enter cells and exert its effects.
That's the crucial bit then.
That's the part doing the heavy lifting at the cellular level. Now, in women, the interaction is slightly different. LH stimulates the corpus luteum in the ovary to secrete testosterone, as we mentioned. However, under normal circumstances in women, it's actually estradiol and progesterone, the predominant female hormones, that act as the primary inhibitors of LH secretion, not testosterone. This highlights the unique regulatory differences between the sexes.
That's a key difference. So it's not always just testosterone itself that's doing the work. It sounds like it undergoes some pretty significant transformations once it's in the body, which then dictate how it actually functions. Could you elaborate on these metabolic pathways?
You've highlighted a critical nuance, yeah. And this is where the story of testosterone becomes even more fascinating. Testosterone isn't a singular molecule performing all its duties directly. It undergoes crucial metabolic transformations into other compounds, some of which are even more potent, actually, and others that are inactive, just preparing for clearance.
Okay, so it changes form.
Exactly. One of the most important and well-understood transformations is its conversion to dihydrotestosterone, or DHT.
DHT, right. I've heard of that.
It's pivotal. This conversion is catalyzed by an enzyme called 5-reductase. What makes DHT so powerful is that it binds to the androgen receptor, that cellular switch we mentioned, with a much higher affinity than testosterone itself.
Binds tighter.
Much tighter. And not only that, but once bound, DHT also activates gene expression, turns on the cellular machinery more efficiently. So in many tissues, testosterone is actually acting as a prohormone, a precursor to the truly active form, which is DHT.
Wow, okay, so testosterone sets it up, DHT delivers the punch.
That's a good way to put it. And we actually have two main forms of this 5-reductase enzyme, and their distribution in the body is quite telling. Type I 5-reductase is predominantly found in non-genital skin, in the liver, and in bone. In contrast, type 2 5-reductase is found mostly in urogenital tissue, in men think prostate and seminal vesicles, and also in the genital skin of both men and women.
Different locations for the enzyme.
Right. And this specific distribution helps explain why DHT plays such a critical role in certain processes, like the development of male external genitalia during fetal life, and later on in conditions such as benign prostatic hyperplasia, or BPH, in adult men.
The prostate connection.
Precisely. Now, another incredibly significant transformation is the conversion of testosterone to estradiol.
Estradiol, like the main estrogen.
Yes, exactly. Estradiol, the primary estrogen. This process is catalyzed by a complex enzyme called aromatase, or CYP19. Aromatase is present in many tissues throughout the body, including fat, bone, and the brain. What's particularly interesting here is that this conversion accounts for about 85% of the circulating estradiol in men.
85% of estrogen in men comes from testosterone.
That's right. The remaining 15% or so is secreted directly by the tests themselves. So a substantial portion of the estrogen circulating in a man's body actually originates from testosterone being converted.
Mind blown, okay.
It's quite something. Finally, the liver plays a critical role in metabolizing testosterone into biologically inactive compounds. These are molecules like androsterone and etiochalanolone. This hepatic metabolism is essentially the body's way of deactivating these powerful hormones and preparing them for excretion.
Cleaning up, basically.
Exactly. Ensuring that their effects are tightly controlled and that excess hormone is cleared efficiently. Even DHT itself is further metabolized into inactive forms like androsterone, androstenedione, and androstenediol. It ensures a complete and efficient pathway for hormonal regulation and clearance.
Okay, so it's clear that testosterone isn't just one thing acting one way. Now that we understand where it comes from, how it travels, and its transformations, let's explore how it actually exerts its profound influence across the body. How does it communicate with ourselves? What are the mechanisms of action that drive its wide-ranging effects?
That's really the heart of how these hormones work, isn't it? Testosterone achieves its incredibly varied and profound effects in tissues through not just one, but at least three primary mechanisms. Think of it like a conductor with several different instruments at its disposal, each playing a crucial part in the symphony of the body.
Okay, three ways.
Three main ways. First, and most directly, testosterone itself can bind to that specific cellular switch we talked about, the androgen receptor, or AR, which is located inside target cells. That's the classic pathway.
Direct binding. Got it.
Second, and often with greater potency in certain tissues, testosterone gets converted to that more powerful form we just discussed, dihydrotestosterone, or DHT. It's this DHT that then binds to the same androgen receptor. But as we noted, DHT often has a higher affinity and efficacy at this receptor.
Binds tighter, stronger response.
Exactly, meaning it binds more tightly and triggers a stronger response in those specific tissues where that conversion happens readily.
Okay, that's two. What's the third?
And third, and this is the one that often surprises people, testosterone can be converted to estradiol. Once converted, this estradiol then binds to the estrogen receptor, initiating estrogenic effects.
Through the estrogen receptor.
Yeah. So this multi-pathway action means that testosterone's influence is incredibly broad and nuanced. It extends far beyond what many might typically expect, just thinking of it as a male hormone. The key insight here is that when we talk about testosterone's effects, we're actually talking about a trio of actions. Direct AR binding, action via DHT binding the AR, and surprisingly, action via estradiol binding the estrogen receptor.
So it's not a single hammer, but like you said, a multi-tool, often using unexpected mechanisms to shape the body.
Exactly. It's not a single hammer, it's a multi-tool.
That's a powerful way to think about it.
Yeah.
Let's delve deeper then into that androgen receptor you mentioned, the AR. What exactly is it? How does it work on a molecular level? And what are the implications when it doesn't function correctly?
Right. The AR is a crucial player. It's a member of a large family of proteins known as the nuclear receptor superfamily. Specifically, it's classified as NR3A. Structurally, the AR is quite complex. It features several distinct parts. There's an amino terminal domain, which has this polyglutamine repeat. Its variable length can actually influence the receptor's activity. Shorter lengths generally mean increased activity.
Interesting detail.
Isn't it? Then there's a DNA binding domain equipped with two distinctive zinc finger motifs. These are specialized structures allowing it to actually grab onto DNA. And finally, a carboxy-terminal ligand binding domain. That's the spot where testosterone or DHT physically attach, like a key in a lock.
Okay, the docking station.
Pretty much. In a resting state, without any androgen present, the AR typically hangs out in the cytoplasm of the cell, often associated with a heat shock protein complex that keeps it stable and waiting. But when testosterone or DHT binds to that ligand binding domain, it triggers a change, a conformational change. This binding causes the AR to let go of those heat shock proteins. Then, two AR molecules pair up. They form a dimer.
Two together.
This dimerized AR complex then translocates or moves into the nucleus of the cell, which is essentially the cell's command center.
Into the control room.
Right. Once inside the nucleus, this dimerized AR, using its DNA binding domains, latches onto specific sequences of DNA on certain genes. These sequences are aptly named androgen response elements, or AREs. The entire ligand receptor complex then acts as a transcription factor. It recruits other proteins called coactivators. And together, this intricate machinery either stimulates or represses the expression of those target genes, turning them on or off. And this gene regulation is the ultimate mechanism through which androgens exert their wide array of biological processes, everything from muscle growth to hair patterns.
That's an incredibly detailed look at how this molecular switch works.
Yeah.
So, given its centrality, what happens if there are mutations or problems with the androgen receptor itself? What are the clinical implications of those kinds of abnormalities?
Oh, the clinical implications of AR mutations are indeed profound. They really illustrate just how critical this receptor is, right from early development onward. For example, mutations affecting either the hormone binding region or the DNA binding regions of the AR can lead to what we call androgen insensitivity or resistance to testosterone's action. This can start as early as in utero, resulting in incomplete male sexual differentiation and, later on, impaired pubertal development.
The development goes wrong.
It can, yeah. We see a wide spectrum of effects here. The most severe forms can lead to a complete absence of androgen action, clinically manifesting as an individual with a male genetic makeup but entirely female external genitalia. Less severe forms might lead to partial virilization of the external genitalia, meaning they might appear ambiguous. And the mildest forms might even allow normal male development in utero, but still result in issues later, like impaired spermatogenesis, the production of sperm in adulthood.
A whole range of outcomes
A
then.
very wide range. And a very specific and striking example of an AR mutation is seen in Kennedy disease, also known sometimes as spinal and bulbar muscular atrophy. Patients with this condition have an expansion of a specific genetic sequence, a CAG repeat, which codes for the amino acid glutamine at the amino terminus of the AR molecule.
It's an expansion in the gene.
Exactly. This expanded repeat causes a very mild form of androgen resistance, primarily noticeable perhaps as gynecomastia, the enlargement of male breast tissue. But much more significantly, it leads to a progressively severe and debilitating motor neuron atrophy, affecting the nerve cells that control voluntary muscles.
Well, that sounds serious.
It is. And while we understand the genetic cause, the precise mechanism by which this specific AR mutation leads to that neuronal atrophy is still an active area of research. We don't fully understand why it targets those neurons.
Still mysteries to solve.
Always. Furthermore, understanding AR mutations helps explain a crucial aspect of prostate cancer progression. Metastatic prostate cancer, as you know, often initially responds dramatically to therapies that deprive the body of androgens, but frequently it becomes unresponsive over time.
Right, it becomes resistant.
Exactly. Often termed androgen-independent or castration-resistant prostate cancer. It's long puzzled clinicians. What we've learned is that even in these resistant cases, the androgen receptor continues to be expressed and its signaling remains active.
Even without much testosterone.
Yeah, possibly due to new mutations in the AR gene itself that allow it to be activated even with very low or no ligand present, or changes in its co-regulatory proteins that enhance its activity. It's truly fascinating because in some of these resistant cases, tumors will even respond to further androgen depletion, for instance, by using drugs like abiraterone, which inhibit androgen synthesis not just from the testes, but also from the adrenal glands.
Wow, hitting it from another angle.
Right. It just underscores the persistent, complex dependence of prostate cancer on androgen signaling, even when it appears to have escaped standard androgen deprivation.
It still strikes me as counterintuitive, though, that a hormone so classically associated with maleness would exert effects through the estrogen receptor. How does that actually happen in real physiology, and what are the tangible effects we see from this pathway? I mean, really.
Yeah, it's a point that often trips people up, but it's absolutely fundamental to fully understanding testosterone's diverse actions. As we briefly touched on earlier, certain critical effects of testosterone depend entirely on its conversion to estradiol. This conversion isn't spontaneous, it's meticulously orchestrated by that enzyme complex called aromatase, or CYP19, and it's widely distributed in various tissues, fat, bone, brain.
Aromatase, right, the converter enzyme.
That's the one. Let me give you a compelling example that dramatically illustrates this. We know of rare cases of males who have a genetic deficiency in either the CYP19 aromatase enzyme itself or a deficiency in the estrogen receptor that estradiol normally binds to.
So either they can't make estrogen from testosterone, or they can't respond to it.
Precisely. In these individuals, a striking abnormality occurs. The epiphyses, those crucial growth plates at the ends of long bones, do not fuse.
They don't fuse, so they just keep growing.
They keep growing. This leads to indefinite long bone growth, resulting in individuals who grow exceptionally tall, often far beyond typical height ranges. But crucially, these patients also suffer from severe osteoporosis. Their bones are weak and prone to fractures.
Tall but brittle bones.
Yeah. What's truly illuminating is what happens with treatment. When oestradiol is administered to patients with an aromatase deficiency, it corrects these bone abnormalities, their epiphyses fuse, their bone density improves.
So giving them the estrogen they lack fixes it.
Right. However, if you administer estradiol to a patient with an estrogen receptor defect, it has no effect on their bone abnormalities.
Because they can't use the estrogen anyway.
Exactly. This clearly and powerfully demonstrates that it's the estrogen, derived from testosterone, acting through the estrogen receptor that is absolutely essential for normal bone growth and density in men. While bone also expresses the androgen receptor, suggesting testosterone may have direct effects too. Well, the estrogenic pathway is undeniable and critically important for bone.
That's really convincing evidence.
Isn't it? Another fascinating example relates to libido. Studies have shown that if you administer estradiol to a male with a CYP19 deficiency, it can actually increase his libido.
Estradiol increases libido in a man.
In that specific context, yes. It strongly suggests that, for some men, testosterone's effect on male libido is at least partially mediated by its conversion to estradiol. It really challenges the conventional wisdom that it's purely an androgenic effect.
Definitely food for thought.
And perhaps one of the most definitive pieces of evidence for this dual mechanism comes from a landmark study, Finkelstein and colleagues, back in 2013. This research was meticulously designed. They took men and suppressed their natural testosterone production using a GnRH analog. Then they replaced testosterone, but in two groups. One group got testosterone alone. The other group got testosterone along with anastrozole, which is an aromatase inhibitor. It blocks that conversion of testosterone to estradiol.
Okay, so one group could make estrogen, the other couldn't.
Precisely. And the results were revelatory. They found that increases in sexual desire, improvements in erectile function, and decreases in subcutaneous and abdominal fat required testosterone's conversion to estradiol.
Really? Needed the estrogen pathway for those.
Yes. If aromatase was blocked, those improvements didn't happen as robustly, even with testosterone present. However, and this is the key separation, the increases in lean mass and muscle strength did not require this conversion. They occurred whether or not aromatase was inhibited.
Ah. So muscle gain is direct androgen action, but fat loss and sexual function involve estrogen conversion.
That's what the study strongly indicates. It powerfully shows that testosterone acts through distinct pathways. Some effects are mediated by the androgen receptor directly or via DHT, while others absolutely depend on its conversion to estradiol and subsequent action through the estrogen receptor.
Wow.
It makes its physiological role far more intricate and multifaceted than we might have once assumed.
That's a truly brilliant breakdown of its mechanisms. It really shifts our perspective. So these powerful hormones aren't just for puberty or adulthood then. They're shaping us from before we're born, continuously influencing us throughout every stage of life. Let's trace that incredible lifelong journey in more detail now.
It's absolutely an ongoing orchestration right from conception onward. In utero, during gestational development, the role of testosterone is profoundly critical. Around the 8th week of gestation, when the fetal testes begin to secrete testosterone, stimulated by HCG from the placenta, the high local concentrations of this hormone are essential. They act on the nearby Wolffian ducts, which are embryonic structures, stimulating them to differentiate and develop into the male internal genitalia.
Internal parts first.
Right. This includes structures like the epididymis, the vas deferens, and the seminal vesicles. Simultaneously, in the embryonic tissue that will form the external genitalia, testosterone undergoes that crucial conversion to dihydrotestosterone, DHT.
Ah, DHT again.
Yep.
It is this DHT that then drives the development of the distinct male external genitalia, shaping the penis and scrotum. And towards the very end of gestation, an increase in testosterone levels can lead to further growth of the phallus. So right from the start, testosterone is literally molding the developing male.
Incredible detail.
Moving into infancy, we observe that fascinating surge in testosterone secretion by the testes during the first few months of life, that mini-puberty. While this peak is well documented, the precise long-term consequences or physiological implications of this specific burst of testosterone during infancy, well, they're still not fully known. It remains an area of ongoing scientific inquiry.
Still learning about that bit.
Definitely. Then puberty in males is perhaps the most dramatic stage of androgen influence, typically beginning around a mean age of 12 years. It's initiated by a significant increase in the secretion of FSH and LH from the pituitary. It's self-stimulated by increased GnRH from the hypothalamus. That whole axis kicks into high gear.
The cascade starts. Right. This hormonal cascade leads to a surge in testosterone production by the Leydig cells, alongside FSH's effects on the Sertoli cells within the testes. Together, these stimulate the development of the seminiferous tubules, the structures responsible for sperm production, ultimately leading to the capacity for mature sperm production.
Okay, fertility enabled.
And the systemic changes during male puberty are profound, occurring gradually over several years. The external genitalia undergo significant enlargement. The phallus increases in both length and width. The scrotum becomes rugate, gets that characteristic wrinkled appearance. The prostate gland also begins to develop and secrete its fluid components for semen.
Lots of growth.
Lots. On the skin, you see it becoming coarser and oilier due to increased sebum production, which often contributes to the development of acne.
Ah, acne, yeah.
A hallmark of male puberty is the differential growth of sexual hair, starting with pubic and axillary underarm hair, then extending to the lower legs, body, and eventually the development of facial hair. Musculoskeletal changes are also very pronounced. A significant increase in muscle mass and strength, particularly noticeable in the shoulder girdle, and a general decrease in subcutaneous fat, leading to a more defined physique.
Getting stronger, leaner.
In terms of bone development, there's an accelerated epiphyseal bone growth. That's a pubertal growth spurt. This spurt eventually leads to epiphyseal maturation and fusion, which marks the permanent cessation of linear growth. Bones also become thicker and stronger.
The growth spurt, then stopping growing taller.
Exactly. Hematologically, there's increased erythropoiesis, the production of red blood cells, resulting in consistently higher hematocrit and hemoglobin concentrations in men compared to boys or women. The larynx, or voice box, thickens, leading to the characteristic lowering of the voice.
The voice trap.
That's the one. And crucially, the development of libido occurs. Beyond these physical changes, there may also be more subtle psychological and behavioral shifts, including tendencies towards better spatial relations and differing behaviors, sometimes including increased aggression.
So once adulthood is reached, are these changes just maintained, or does testosterone continue to influence new developments or conditions that manifest later in life?
That's a great question. During early adulthood and midlife, the serum testosterone concentration and all those adult male characteristics established during puberty are largely maintained. It forms a sort of stable baseline. However, testosterone continues to play a role in new developments. One common and familiar change that typically begins to develop during this phase is male pattern baldness, often starting with recession at the temples or the vertex, the crown of the head.
Ah, yes, hair loss.
Yep. And beyond appearance, two significant medical conditions are very closely linked to androgen action in adulthood. The first is benign prostatic hyperplasia, or BPH. This condition affects almost all men to varying degrees as they age. It involves the non-cancerous enlargement of the prostate gland, which can sometimes obstruct urine outflow by compressing the urethra as it passes through the prostate.
BPH. Very common.
Very common. And crucially, the development of BPH is primarily mediated by the conversion of testosterone to dihydrotestosterone, DHT, specifically by that 5-reductase type 2 enzyme within the prostatic cells.
GHG again, driving prostate growth.
Exactly. The second condition, and a very serious one, is prostate cancer. Now, while it's important to clarify that there is no direct evidence that testosterone causes prostate cancer.
Right, not causation.
Not causation proven. But the disease is definitively dependent on androgen stimulation for its growth and progression. This fundamental dependency is what forms the entire basis for how we treat metastatic prostate cancer, by either aggressively lowering serum testosterone concentrations or by blocking its action at the androgen receptor within the cancer cells.
Using its dependency against it.
Precisely. It highlights how manipulating androgen pathways is a cornerstone of prostate cancer therapy. Finally, as men transition into senescence, or old age, there's that characteristic gradual decline in serum testosterone concentration. Simultaneously, there's often a gradual increase in the concentration of sex hormone-binding globulin, SHBG.
So less free testosterone available again.
Exactly, less free active testosterone. By the age of 80, for instance, a man's total testosterone levels might be around 80% of what they were at age 20, but his free testosterone, the biologically active portion, could be as low as 40% of his youthful levels.
It's a big drop in the active form.
It is. And the significant age-related fall in testosterone is believed to contribute to several other common changes that occur with increasing age in men. Things like decreases in libido, a reduction in bone mineral density, and lower hemoglobin levels. We see evidence supporting this link in studies where normalizing testosterone levels in older men has led to improvements in these very parameters. So testosterone's influence is truly continuous and multifaceted, shaping us from before birth to the very latest stages of life.
That journey across the lifespan really emphasizes how critical testosterone is. So what happens if something goes fundamentally wrong with testosterone production or action before we're even born? The consequences sound like they could be incredibly profound, literally setting the stage for lifelong differences in development.
Oh, absolutely. The role of testosterone during fetal development is, without exaggeration, absolutely critical for male sexual differentiation. If a male fetus experiences testosterone deficiency during that crucial first trimester, it leads to incomplete sexual differentiation. In the most severe cases, a complete deficiency of testosterone secretion at this early stage can result in entirely female external genitalia, despite the fetus being genetically male, XY.
Wow, female appearance despite male genes.
Yes. This deficiency also means the Wolffian ducts, those embryonic precursors to male internal genitalia, fail to differentiate into structures like the epididymis and vas deferens. However, it's an important distinction that the Mullerian ducts, which would normally form female internal genitalia, do not differentiate into female internal genitalia in this scenario.
Why not?
Because the testes, even if not making enough testosterone, are usually still present and secreting Mullerian inhibitory substance, or MIS. MIS actively suppresses female internal organ development, so you end up with neither fully developed male nor female internal structures in the most severe cases.
Complex outcomes.
Very complex. And what's equally fascinating and sometimes more challenging to diagnose is that similar outcomes can occur even if testosterone is secreted normally, but its action is diminished due to an abnormality of the androgen receptor itself or that reproductive enzyme.
So the signal is sent, but not received properly.
Precisely. Abnormalities of the androgen receptor, the AR, can have an incredibly wide spectrum of effects, showcasing its intricate role. The most severe forms of AR abnormalities result in a complete absence of androgen action, leading to that female phenotype we mentioned. On the other end, moderately severe forms lead to partial virilization of the external genitalia, which might appear ambiguous. And the mildest forms might actually permit normal virilization in utero, but still result in impaired spermatogenesis, the ability to produce sperm in adulthood.
Again, that spectrum of severity.
Exactly. Now, if the issue lies specifically with an abnormal reproductive enzyme,
The DHT converter enzyme.
right. If that's deficient, it results in incomplete virilization of the external genitalia during fetal development because they need DHT. However, because the male internal genitalia, like the epididymis and vas deferens, only require testosterone itself for their development, not its conversion to DHT, these internal structures develop normally.
Okay. Internal, okay. External affected. That's a key clue.
It's a key diagnostic clue for that specific deficiency. Testosterone deficiency during the third trimester of pregnancy has a more focused impact. It primarily impairs phallus growth, leading to a condition known as microphallus, or an abnormally small penis. This finding is quite common in boys who are later found to have abnormalities in LH secretion, often due to underlying problems with GnRH secretion or action from the hypothalamus.
Linked to pituitary issues later on.
Often, yes. Additionally, significant testosterone deficiency, particularly during fetal development, can result in the testes failing to descend into the scrotum, a condition called cryptorchidism. This is commonly observed in boys with subnormal LH secretion, further underscoring the interconnectedness of these hormonal pathways from the earliest stages.
So what if a boy is able to produce testosterone normally in the womb, allowing for that initial male development, but then perhaps due to a medical condition or injury, loses that ability before puberty is complete? What are the specific impacts and consequences in that critical developmental window?
Yeah, that's a crucial scenario with distinct consequences. If a boy secretes testosterone normally in utero, allowing for male sexual differentiation, but then loses that capacity before the anticipated age of puberty, the primary result is a failure to complete puberty. All those hallmark pubertal changes we discussed earlier, the development of external genitalia, the appearance of sexual hair, the increase in muscle mass, the deepening of the voice, the behavioral shifts, they'll all be impaired.
Just stops mid-process.
Essentially, yes. The degree of impairment will directly correspond to how abnormal the testosterone secretion is. If it's a complete absence, these changes simply won't occur. If it's partial, they'll be stunted or delayed.
Depends on how much is missing.
Right. Furthermore, if growth hormone secretion remains normal, but testosterone levels are subnormal during those critical years of expected puberty, a very specific skeletal change occurs. The long bones in the arms and legs will continue to lengthen because the epiphyses, the growth plates, don't receive the necessary hormonal signal from testosterone to close.
So they keep growing longer.
They do. This leads to a distinct body proportion. Individuals will have disproportionately longer arms and legs relative to their trunk. It's a classic finding in untreated prepubertal hypogonadism, sometimes called unicoid proportions.
Okay, I see. Long limbs.
And another common consequence of subnormal testosterone secretion during the age of expected puberty is the enlargement of glandular breast tissue, a condition medically termed gynecomastia.
Gynecomastia again. Why does that happen?
It occurs because even with low testosterone, there might still be some estrogen activity, or the ratio of estrogen to androgen becomes skewed, leading to unopposed estrogenic stimulation of breast tissue development. These prepubertal deficiencies truly highlight testosterone's orchestrating role in the entire pubertal symphony.
So we've talked about early life deficiency. What if testosterone levels drop later in life, perhaps due to medical reasons, injury, or simply the aging process we discussed?
How quickly and in what ways do those established pubertal effects regress once testosterone is no longer optimally present?
That's a common question. The regression of testosterone's effects after puberty is completed, well, it depends quite a bit on both the degree and the duration of the deficiency. It's not usually a sudden reversal. Some effects recede relatively quickly, while others diminish much more slowly, almost imperceptibly over many years.
So it varies.
It varies. For instance, if the deficiency is substantial, say, due to a medical condition that severely impairs testicular function, some of the most sensitive, non-physical effects will be noticed quite quickly. Changes in libido or sex drive and overall energy levels typically decrease within just a week or two of the deficiency becoming pronounced. These are often the first signs that people notice.
Libido and energy first.
Often, yeah. Other testosterone-dependent characteristics decline more gradually. A noticeable decrease in hemoglobin, the oxygen-carrying protein in red blood cells, will usually become apparent within several months. Bone mineral density, which testosterone helps maintain, can be detected as decreasing within about a year of deficiency.
A year for bone density.
To start showing measurable decline, yes. But it's important to note that an increase in the actual risk of bone fracture isn't likely to occur for many, many years after that initial bone density decline starts. Interestingly, a clinically detectable decrease in muscle mass in an individual doesn't typically happen for several years after testosterone levels fall.
Muscle loss takes years. Huh.
It does. And the loss of sexual hair, facial hair, body hair, also takes many years to become noticeable. So while some immediate subjective changes occur, many of the physical regressions are a much slower burn.
Got it. Gradual for many physical things. Now, while our conversation is largely focused on men, it's important we circle back to women. Androgens, including testosterone, play a role in women too. What are the consequences when women experience androgen deficiency?
That's a vital point, absolutely. Androgens are indeed important in women, though present in much lower concentrations compared to men. When there's a loss of androgen secretion in women, the most consistently observed and noticeable effect is a decrease in sexual hair, think pubic and axillary hair. However, similar to the long-term effects in men, this change is very slow and often takes many years to become apparent to an individual.
A slow hair loss again.
Pretty much. Beyond hair, androgens may have other important, though perhaps less clearly defined or extensively studied, effects in women. For instance, a severe loss of ovarian and adrenal androgens, which can occur in conditions like panhypopituitarism, where the pituitary gland isn't producing enough stimulating hormones, may lead to a constellation of symptoms. These could include a decrease in libido, reduced energy levels, a loss of muscle mass and strength, and a decline in bone mineral density.
Similar symptoms to men, potentially.
Potentially, yes. But the research data specifically on androgen deficiency in women and its treatment are not as robust or extensive as they are for men. It's definitely an area where more understanding is still needed. But the evidence suggests that even in lower concentrations, these hormones contribute meaningfully to female physiology and well-being.
Okay, good to cover. So if someone does have androgen deficiency, whether male or female, how do we actually go about replacing it? It sounds like it's not as simple as just popping a pill and expecting it to work effectively, given everything we've learned about its metabolism and transport.
You're absolutely right. It's far from simple. Ingesting testosterone itself, orally, is largely ineffective for therapeutic replacement. Why? Because it undergoes rapid hepatic metabolism, that first pass event we talked about, after it's absorbed from the intestine.
The liver breaks it down too fast.
Exactly. Most of the testosterone would be broken down by the liver before it could even reach the systemic circulation and exert its desired effects throughout the body. That's a significant pharmaceutical challenge. And it's also crucial to highlight that all FDA-approved testosterone, methyltestosterone, and oxandrolone products are classified as Drug Enforcement Administration Schedule 3 controlled substances.
Schedule 3, so controlled substances.
Yes. This designation underscores their potency, their potential for misuse and abuse, and the need for strict medical oversight.
Right. So how do we get around that liver problem?
Well, to ingeniously bypass this rapid hepatic metabolism, pharmaceutical scientists have developed various approaches over the years. One of the oldest and still widely used methods involves creating testosterone esters. These are formed by attaching a fatty acid like enanthate, cypionate, or undecanoate to the 17-hydroxyl group of the testosterone molecule. This modification makes the testosterone more lipophilic, or fat-soluble.
Making it oilier, sort of.
Basically, yes. When an ester like testosterone enanthate or cypionate is dissolved in an oil solution and administered intramuscularly injected into the muscle, typically every 1 to 2 weeks, the ester slowly hydrolyzes in the body, gradually releasing the native active testosterone over time.
Okay, slow release from the injection site.
That's the idea. However, a drawback with these biweekly injections is that serum testosterone concentrations can fluctuate quite widely. They might be higher than normal in the first few days after the injection, then gradually decline to low normal levels just before the next injection is due. Sort of a peak and trough effect.
A roller coaster.
A bit, yeah. And attempts to reduce the frequency of injections by simply increasing the dose tend to lead to even wider fluctuations and can result in poorer clinical outcomes for the patient. More isn't always better here.
Consistency is key then.
Ideally, yes. Now, testosterone undecano is another ester, but it's unique. When dissolved in oil and ingested orally, it's designed to be absorbed predominantly into the lymphatic circulation.
Lymphatic, not the main blood vessels.
Right. This clever route of absorption allows it to largely bypass that initial destructive hepatic first-pass metabolism, making oral administration effective for this specific ester. It's also available as an injectable formulation, and this particular injectable version is notable because it produces remarkably stable and sustained serum testosterone concentrations for up to 2 months.
2 months from one shot.
Wow. Yeah, offering a much more consistent level than the more frequent biweekly injections. It's a significant advance for some patients.
Definitely sounds more convenient. What other options are there?
Another class of agents is the 17 alkylated androgens. These include compounds like methyltestosterone, oxandrolone, stanozolol, fluoxymesterone, and danazol. In these molecules, an alkyl group, a small carbon chain, is added to the 17 position. This modification is specifically designed to retard their hepatic metabolism, slow it down.
So the liver doesn't break them down as fast, allowing oral use.
Exactly. It enables effective oral administration. However, this convenience comes with significant trade-offs. A key issue is that these compounds are generally less androgenic in their overall effect compared to native testosterone. More importantly, they carry a notable risk of hepatotoxicity or liver damage.
Liver problems.
Yes. This can manifest as cholestasis, a reduction in bile flow, uncommonly as pileosis hepatis, a rare condition with blood-filled cysts in the liver, and very rarely as hepatocellular carcinoma, a type of liver cancer.
Yikes. Serious risks.
Definitely. While some of these, often termed anabolic steroids, were initially thought to exhibit greater anabolic muscle building than androgenic male characteristic promoting effects in lab rats, well, this differential effect has not been convincingly demonstrated in humans. Indeed, the FDA has issued strong warnings against their use in bodybuilding products due to these serious health risks.
So the benefit-risk profile is tricky for those.
Very tricky, which led to other approaches. To avoid hepatic first-pass inactivation and provide more stable serum testosterone concentrations, transdermal delivery systems become very popular. These include gels or solutions that are applied to the skin, or sometimes the nasal mucosa, transdermal patches that adhere to the skin, and even buccal tablets that are placed against the gum.
Gels, patches, delivering it through the skin.
Right. These systems are ingeniously designed to deliver native testosterone directly into the systemic circulation through the skin or mucous membranes. They often utilize specific chemical enhancers or excipients to facilitate controlled absorption across these barriers. And the goal is providing a more consistent and physiological serum testosterone profile compared to the fluctuating levels seen with some injections.
Trying to mimic the body's natural rhythm more closely.
That's the aim. Finally, there's been significant research into selective androgen receptor modulators, or SARMs.
SARMs, right. Heard about those too. Often not in a medical context.
Unfortunately, yes. These are non-steroidal compounds that have been engineered with a very specific goal: to exhibit desirable testosterone-like effects in certain tissues, like muscle and bone, while ideally avoiding undesirable effects in other tissues, like the prostate.
Selective action. Get the good, avoid the bad.
That's the hope, to selectively activate the androgen receptor only where it's beneficial. Some SARMs have been developed and tested in human clinical trials, offering promise for future therapeutic options. However, unlike selective estrogen receptor modulators, or SERMs, which are used clinically, well, none of these SARMs are currently approved for clinical use.
Not approved yet.
Not approved, despite some unapproved compounds being regrettably available illicitly online. So while the concept is powerful, actual clinical application is still on the horizon for SARMs.
That's a comprehensive overview of how we get testosterone into the body. Now let's turn to the why. It sounds like there are very specific and often crucial situations where therapeutic androgens become incredibly important tools in medicine. Let's delve into those established and maybe emerging uses, and importantly, how they're monitored.
Absolutely. The best established and most clear-cut indication for androgen administration is undeniably testosterone replacement therapy for male hypogonadism, which just means a deficiency in testosterone. The primary treatment goal here is straightforward: to restore and mimic normal physiological serum testosterone concentrations. Any of the preparations we just discussed can potentially be effectively used to achieve this.
So just getting levels back to normal.
That's the core goal. Monitoring the efficacy of this treatment is absolutely crucial. The most important aspect is regularly measuring the serum testosterone concentration during treatment. For transdermal gels, which are designed to provide relatively constant levels, measurements can often be taken at almost any time. For the biweekly testosterone esters, like enanthate or cypionate, the ideal time to measure is usually midway between doses.
Mid-cycle for the shots.
Right. And if that midway measurement isn't within the normal range, the dosage likely needs to be adjusted. Additionally, if the testosterone deficiency is due to primary testicular disease, which is usually indicated by an elevated serum LH level before treatment, then a normalization of LH levels within about 2 months of starting treatment is an indirect but reassuring sign that the testosterone replacement is adequate.
LH coming down means the body recognizes enough testosterone is present.
Exactly, the feedback loop is responding. The outcomes of normalizing serum testosterone in hypogonadal men are quite significant and often profoundly improve quality of life. In prepubertal boys with hypogonadism, testosterone replacement induces normal virilization, allowing them to go through puberty properly. In adults who became hypogonadal after puberty, it restores those virilized characteristics.
Brings things back to normal.
Right. Some improvements are quite rapid. Libido, overall energy levels, and hemoglobin concentrations typically return to normal within just a few months. Body composition also shifts favorably, with increases in muscle mass and decreases in fat mass generally observed within about 6 months.
Quicker results for some things.
Yes. Other benefits take longer to accrue. Bone density and trabecular connectivity, that internal architecture of bones, they continue to increase for up to 2 years of consistent treatment.
Bone health takes longer.
Okay. It's a slower process. However, alongside these benefits, we must meticulously monitor for potential deleterious effects or side effects. At physiological doses, especially with transdermal delivery, there are generally few side effects beyond those of endogenous testosterone itself, unless the dose becomes excessive. But if doses are indeed excessive, they can mirror the accelerated changes of puberty, leading to things like acne, gynecomastia, or even more aggressive sexual behavior.
Too much of a good thing.
Precisely. Even without a predisposition, excessive doses can uncommonly lead to erythrocytosis, that increase in red blood cell count, making blood thicker. And in some cases, salt and water retention, potentially causing peripheral edema or swelling. For men over 40, there's a particular concern regarding those testosterone-dependent diseases that can emerge later in life, specifically benign prostatic hyperplasia, BPH, and prostate cancer. Monitoring for those is key.
Prostate checks, PSA levels, and such.
Absolutely. Regular monitoring is standard practice. And it's crucial to remember that the modified testosterones, particularly those 17 alkylated ones we discussed earlier, carry specific side effects even at physiologic doses. Their primary concern is hepatic toxicity cholestasis, pileosis hepatis, rarely liver cancer. Furthermore, these 17 alkylated compounds can negatively impact serum lipids, decreasing good HDL cholesterol and increasing bad LDL cholesterol, especially at larger doses.
So those oral alkylated ones need extra liver monitoring.
Definitely need careful liver function monitoring. Now, when treating boys at the anticipated time of puberty, a very important consideration is height and growth. Testosterone accelerates epiphyseal maturation. It initially leads to that characteristic pubertal growth spurt, yes, but it also ultimately leads to the closure of those growth plates, permanently ceasing linear growth.
Stops them growing taller eventually.
Right. Therefore, a boy's current height and his growth hormone status must be carefully considered. If a boy is short primarily due to growth hormone deficiency, he should ideally be treated for that first, before initiating testosterone treatment for hypergonadism to maximize his final adult height potential.
Timing is important for height.
Very important. Moving beyond classical hypogonadism, there's the complex area of male senescence or aging men. As men age, serum testosterone declines, and this decline often parallels other age-related changes like decreased sexual function, reduced bone density, lower hemoglobin levels. This parallel suggests that the age-related fall in testosterone likely contributes to these observed changes.
The low T discussion in older men.
Exactly. A significant set of studies, often referred to as the T trials, involving nearly 800 men aged 65 or older with low testosterone, provided valuable insights. They found that 1 year of testosterone treatment compared to a placebo significantly improved sexual function, mood, depressive symptoms, anemia, bone density, and muscle strength.
That's pretty positive.
It does show benefits for those specific things. However, it's important to note that it did not improve cognitive function or glucose and lipid metabolism in those trials.
Didn't help everything.
Didn't help everything. And a critical point here, with significant regulatory implications, is that no studies to date have been large enough or long enough to definitively determine if testosterone treatment in older men increases the long-term risk of prostate cancer, worsening urinary tract symptoms from BPH, or cardiovascular disease.
The long-term safety data isn't fully there yet.
Especially regarding cardiovascular risk. Due to ongoing concerns, some based on epidemiological studies and smaller trials, the FDA has mandated label changes for testosterone preparations. These now explicitly approve testosterone products only for classical hypogonadism, meaning a discernible pituitary or testicular disease, and not for idiopathic or unexplained or purely age-related hypogonadism.
So only for diagnosed medical conditions, not just aging.
That's the current FDA stance, yes. It remains a very active and debated area in medicine.
Understood. What about female hypogonadism? You mentioned the data is more limited.
Considerably more limited compared to men, yes. There are few robust studies on whether systematically increasing serum testosterone in women with low levels improves symptoms like low libido, reduced energy, decreased muscle mass strength, or diminished bone mineral density. One small study, Miller and colleagues in 2006, involved women with low testosterone due to panhyperpituitarism. It did find that increasing their testosterone levels to high normal ranges was associated with small but statistically significant increases in bone mineral density, fat-free mass, and sexual function compared to placebo.
Small increases, okay.
Small but measurable. However, the overall evidence base is just not as strong, and more research is definitely needed to clearly define the role and safety of testosterone replacement in women.
More work needed there. Now let's touch on the elephant in the room. The illicit use of androgens for athletic performance enhancement.
Right. The non-medical use. Information here is inherently less complete than for therapeutic uses, obviously, given its clandestine nature. The FDA has repeatedly warned against bodybuilding products marketed as containing steroids due to the serious health risks involved.
Understandably.
Virtually all androgens produced for human or veterinary use have, at some point, been abused by athletes. Initially, those 17 alkylated androgens, the anabolic steroids, were favored partly due to a perceived greater anabolic effect, whether real or not, in humans. Now, partly to evade detection, athletes often turn to testosterone esters or even human chorionic gonadotropin, HCG, which boosts the body's own testosterone production.
Trying to look more natural to tests.
Sometimes, yes. Even testosterone precursors like androstenedione and DHEA, sometimes marketed wrongly as nutritional supplements, are used. There are also those unapproved selective androgen receptor modulators, SARMs, regrettably available online. And famously, tetrahydrogesterone, or THG, a potent androgen, was specifically designed by Belco to evade detection due to its novel chemical structure and rapid catabolism. The designer steroid.
Right, THG. So, do they actually work for performance?
Regarding efficacy for athletic performance, controlled studies, despite the ethical challenges, do suggest a clear dose-dependent effect of testosterone on muscle strength, which acts synergistically with exercise. A landmark study by Bosson and colleagues in 1996 showed that superphysiological doses of testosterone and enanthate, we're talking 600 milligrams per week, more than 6 times a typical replacement dose, significantly increase muscle size and strength in normal young men, with even greater increases when combined with strength training.
Huge doses, big effects.
Yes. A subsequent study by Bosson in 2001 further confirmed this dose-dependent effect of testosterone on muscle strength in healthy young men. In contrast, a study on oral androstenedione found that it did not lead to an increase in muscle strength or mean serum testosterone, highlighting that not all these compounds are equally effective, especially precursors.
So testosterone itself at high doses works, precursors maybe not so much.
Seems that way from the limited controlled data. But these high doses for performance enhancement carry significant risks and side effects. Almost all androgens, when taken in high doses, suppress gonadotropin secretion, LH and FSH. This, in turn, suppresses endogenous testicular function.
Shuts down natural production.
Completely, leading to decreased natural testosterone and sperm production, resulting in diminished fertility. While fertility usually returns within about 4 months after stopping, it can sometimes take over a year. Testicular size may also diminish with long-term use.
Fertility issues, shrinking testes?
Yes. Erythrocytosis, that increase in red blood cells, is also common. Gynecomastia, male breast enlargement, is often caused by androgens that can be converted to estrogens, like testosterone itself, but generally not by non-aromatizable androgens like DHT. Those 17 alkylated androgens, again, are unique in their risk of hepatotoxicity and their negative impact on serum lipids, decreasing HDL, increasing LDL.
Liver and cholesterol problems specific to those.
Right. And in women and children, virilization is a profound risk. Facial and body hirsutism, male pattern hair recession, acne, enlargement of the clitoris or phallus. For boys and girls with open epiphyses, these high doses cause premature closure of growth plates and stunting of linear growth, permanently limiting their adult height.
Permanent height reduction.
Yeah.
Serious consequences.
Extremely serious. Detecting illicit use is a constant cat-and-mouse game for anti-doping agencies. For non-testosterone androgens, detection relies on techniques like gas chromatography and mass spectroscopy, identifying the drug or its metabolites if taken recently. For exogenous testosterone itself, it's trickier. One method involves the TE ratio, measuring the ratio of testosterone glucuronide to its endogenous epimer, epitestosterone glucuronide, in urine.
Testosterone versus epitestosterone.
Exactly. Exogenous testosterone suppresses both endogenous T and E, but it only replaces T, thus increasing the TE ratio. However, this method has limitations due to genetic variations in a key enzyme, which can affect epitestosterone excretion naturally.
Not foolproof.
It's not foolproof. A more sophisticated and definitive technique is gas chromatography combustion isotope ratio mass spectrometry, or GCC-IRMS. This looks at the ratio of carbon-13 to carbon-12 isotopes. Urinary steroids with a lower 13C-12C ratio are indicative of a pharmaceutical exogenous source, as plant-derived precursors used in manufacturing have less C13 than what the body makes endogenously.
Wow, isotope ratios. That's high-tech. Much harder to fool.
Much harder. Beyond performance enhancement, androgens have been explored for specific medical conditions too, for catabolic and wasting states. Well, despite testosterone's undeniable anabolic muscle-building effects, it is generally not proven effective for ameliorating general catabolic and muscle-wasting states across the board.
Doesn't work for general wasting.
Generally not. A notable exception, however, is muscle wasting associated with AIDS, especially when it's accompanied by hypogonadism. Treatment of HIV-infected men experiencing AIDS-related muscle wasting and who also have subnormal testosterone levels has been shown to increase both their muscle mass and strength.
Specific case for AIDS wasting.
Another specific use is in angioedema. This is a rare, potentially life-threatening disease caused by impaired C1 esterase inhibitor function, leading to episodes of severe swelling. Chronic androgen treatment, specifically with 17 alkylated androgens like stanozol or danazol, has proven effective in preventing attacks by stimulating the liver to synthesize more of this crucial inhibitor.
Helping prevent swelling attacks.
Exactly. But again, the trade-offs. Virilization is a potential side effect in women. In children, the risk of virilization and premature epiphyseal closure prevents chronic use, though they might occasionally be used for acute episodes. Fortunately, concentrated C1 esterase inhibitor derived from human plasma is now available as a safer alternative for long-term protection.
Better options now.
Much better. Lastly, regarding blood dyscrasias, disorders of the blood, androgens were historically used to stimulate erythropoiesis, red blood cell production, in various forms of anemia. However, this use has largely been supplanted by the availability of erythropoietin, or EPO, a more targeted and effective treatment.
TPO replaced it mostly.
Mostly. Despite this, androgens like danazol are still occasionally used as an adjunctive treatment for certain types of hemolytic anemia and idiopathic thrombocytopenic purpura, particularly those that are refractory or unresponsive to first-line agents. These examples really showcase the versatility, but also the very specific indications and risks for these powerful hormones in medicine.
That's a really thorough look at the uses and abuses. Now, let's explore the other side of the coin completely.
Sure.
What if someone has too much androgen activity, or if we medically need to reduce its influence for a specific condition, like prostate cancer? How do we turn down the tap on testosterone secretion itself?
Right. This is where the class of drugs known as antiandrogens comes into play, targeting excessive androgen action. If the goal is to inhibit the body's own production of testosterone, one primary and very effective approach involves using GnRH analogs.
GnRH analogs like
Yeah.
leuprolide.
Exactly. Leuprolide, goserelin, triptorelin, histrelin, buserelin, those are common examples. When these analogs are administered repeatedly, they initially cause a surge, paradoxically, but then they lead to a downregulation, a sort of desensitization of the GnRH receptors in the pituitary gland.
Overstimulates, then shuts down the receptor.
Precisely. This effectively inhibits the secretion of LH from the pituitary, and as a direct consequence, testosterone secretion from the testes is dramatically reduced, sometimes to castration levels. Their primary and very effective clinical use is in the treatment of prostate cancer, where reducing androgen levels is a cornerstone of therapy.
Chemical castration, essentially.
Medically speaking, yes, that's the effect. Another class of drugs that can inhibit testosterone synthesis, though generally not used as a primary treatment for androgen excess due to their side effect profiles, are the imidazole antifungals, such as ketoconazole.
Ketoconazole,
Yeah.
the antifungal.
Yep. These drugs act by inhibiting various cytochrome P450 enzymes. These CYP enzymes are crucial for the synthesis of many steroid hormones. So by blocking them, ketoconazole can inhibit the synthesis of not only testosterone, but also cortisol.
Hits cortisol, too. That sounds risky.
It is. While sometimes employed in specific cases of glucocorticoid excess, too much cortisol, their use for androgen synthesis inhibition is limited due to the risks of inducing adrenal insufficiency, where the body can't produce enough stress hormones, and also hepatotoxicity, liver damage. So while they can turn down the tap, it comes with significant cautions.
So if for some reason we can't or shouldn't stop the body from making testosterone, or we need to block androgens from other sources like the adrenals, are there strategies to block its effects once it's already circulating? How do we prevent androgens from doing their job at the cellular level?
Absolutely. If we can't inhibit the synthesis or secretion, or if the goal is to block the effects of androgens coming from other sources, we can use drugs that interfere with androgen action at the receptor level or block their conversion to more potent forms. These fall primarily into two categories: androgen receptor antagonists and 5-reductase inhibitors.
Blocking the receptor or blocking the conversion to DHT.
Exactly. Androgen receptor, or AR, antagonists are designed to directly inhibit the binding of androgens like testosterone and DHT to the androgen receptor itself. They sit in the receptor and block the key from fitting. This class includes drugs such as flutamide, bicalutamide, nalutamide, and newer, often more potent agents like enzalutamide, apalutamide, and darolutamide.
The wides.
Many-endinutamide, yes. However, when these drugs are used by themselves, their efficacy for lowering overall androgen effect can be limited,
Why is that?
because the body's negative feedback loop kicks in again. Blocking the AR can be sensed by the brain, causing LH secretion to increase, which then stimulates the tests to produce higher serum testosterone concentrations. This surge can partially overcome the receptor block, like trying to plug a leak while turning the tap on harder.
The body fights back.
It tries to compensate. Therefore, AR antagonists are primarily used in conjunction with a GnRH analog in the treatment of metastatic prostate cancer. This combination therapy is incredibly powerful. The GnRH analog reduces testicular testosterone production way down, while the AR antagonist simultaneously blocks the action of any remaining androgens, including those originating from the adrenal glands, which are not inhibited by GnRH analogs alone.
A double blockade.
Yeah.
Hits testicular and adrenal androgens.
Right, a comprehensive blockade. On a different note, flutamide, one of the older ones, has historically been used off-label to treat hirsutism, excessive hair growth in women, but its association with hepatotoxicity means it's used with significant caution, particularly for cosmetic purposes.
Liver issues, again with flutamide.
Yes. Beyond the dedicated AR antagonists, we have spironolactone. Remember that one?
Yeah, the diuretic.
Primarily known as a diuretic and an inhibitor of aldosterone, yes. But spironolactone also surprisingly acts as a weak inhibitor of the androgen receptor and a weak inhibitor of testosterone synthesis. Interestingly, gynecomastia, male breast enlargement, is a common side effect in men taking spironolactone for conditions like fluid retention or hypertension. This side effect was actually a key driver behind the development of eprenone, a more selective mineralocorticoid receptor antagonist, specifically to avoid this antiandrogenic effect.
So they made a cleaner version.
Essentially. In women, however, spironolactone can be effectively used to treat hirsutism, leveraging that antiandrogen effect. Another agent, cyperterone acetate, is a progestin that also functions as a weak antiandrogen by binding to the AR. It's moderately effective in reducing hirsutism either alone or combined with an oral contraceptive, but it's important to note that it is not approved for use in the U.S.
Okay. Now what about the other approach, blocking the conversion?
Right, the 5-reductase inhibitors. This second major class of drugs that inhibit androgen action includes finasteride and dutasteride.
Finasteride for hair loss and prostate.
Exactly. These drugs are antagonists of the 5-reductase enzyme. They block that crucial conversion of testosterone to the more potent dihydrotestosterone, or DHT. This action is particularly important in tissues like the male external genitalia, hair follicles, and especially the prostate, where DHT is the primary active androgen driving growth.
So they lower DHT levels specifically.
Precisely. These inhibitors were specifically developed and are approved in the U.S. and many other countries to treat benign prostatic hyperplasia, BPH. When administered to men with moderate to severe symptoms resulting from urinary outflow obstruction caused by an enlarged prostate, these drugs significantly decrease both serum and prostatic DHT concentrations. This reduction leads to a shrinking of prostatic volume and a measurable increase in urine flow rate, alleviating those bothersome urinary symptoms.
Helps shrink the prostate, improve flow.
That's the main goal in BPH. In terms of side effects, sexual side effects like decreased libido or erectile dysfunction are documented, though generally infrequent. Gynecomastia is a rare occurrence. Beyond BPH, finasteride at a lower dose is also approved for the treatment of male pattern baldness because DHT is involved there too, and it can be effective off-label in treating hirsutism in women, again showcasing the targeted power of blocking DHT production.
Very interesting how targeting that one enzyme has multiple applications. Okay, let's shift gears now from the broader scope of androgen action and inhibition to a very specific yet incredibly common challenge in male reproductive health, erectile dysfunction, or ED. To truly understand how we treat it, we first need to really unpack the incredibly intricate, multi-layered process of erection itself. It's much more than just a simple physical response, right?
You're absolutely right. Understanding the mechanics of normal erectile function is key to comprehending its dysfunction and how our medications intervene. It's quite fascinating, actually. Normal erectile function is a sophisticated interplay of visual cues, psychological factors, hormonal signals, including adequate testosterone, and neurological impulses. All these diverse factors, from a romantic thought to direct physical touch, ultimately converge on a common underlying mechanism within the penis, increasing the synthesis of a crucial signaling molecule called nitric oxide, or NO.
Nitric oxide, okay, the key player.
It's the initial trigger. Nitric oxide is produced by the vascular endothelium, the inner lining of blood vessels, specifically in the arterioles, the small arteries, supplying the corpora cavernosa. Those are the sponge-like erectile tissues within the penis. NO is also produced by nerve endings within the corpora cavernosa themselves. Once produced, NO diffuses very rapidly to adjacent smooth muscle cells surrounding these blood vessels and within the erectile spaces.
Spreads the signal.
Right. This diffusion triggers a cascade that causes vasodilation, meaning the arterioles widen, allowing much more blood to flow into the penis. Simultaneously, it increases the compliance or relaxation of the cavernosal smooth muscle itself, enabling it to engorge rapidly and significantly with blood.
More blood in, space relaxes to hold it.
Exactly. And this accumulation of blood also plays a crucial dual role. It then physically restricts venous outflow by compressing the small veins against the surrounding tunica albuginea, which is the tough fibrous sheath encasing the corpora cavernosa.
It traps the blood inside.
Precisely. This combined effect, massively increased inflow and significantly restricted outflow, is what ultimately leads to and maintains penile erection.
Makes sense. So what goes wrong in ED?
Well, erectile dysfunction, therefore, can arise from a wide variety of factors that disrupt any part of this intricate process. These include psychological issues like stress or anxiety, hormonal imbalances such as low testosterone, and very commonly vascular causes, often involving damage to the endothelium, which impairs crucial nitric oxide production. It can also be a side effect of various drugs, including some common medications used for hypertension or depression. And it's frequently associated with systemic conditions like diabetes or heart disease, which can damage both blood vessels and nerves involved in the process.
Many potential causes then.
A wide range.
So it's a very delicate dance of signals. Let's delve even deeper into that intracellular signaling pathway you mentioned. How does nitric oxide actually trigger this relaxation and blood flow at the molecular level? And where does that crucial enzyme, PDE5, fit into this whole elegant process?
Okay, yeah, let's zoom in. This is where the molecular magic happens, inside the smooth muscle cells. The nitric oxide, once produced, acts by binding to and activating an enzyme called soluble guanylyl cyclase, often abbreviated as SGC. When SGC is activated by NO, it catalyzes a fundamental reaction. It produces cyclic GMP, or cGMP, from the cell's readily available GTP.
CDMP. Okay, the messenger molecule.
Think of cGMP as the critical second messenger in this pathway. It's the molecule that truly carries the relax signal forward within the cell.
Got it. NO makes cGMP. What does cGMP do?
Good question. Cyclic GMP then activates another key enzyme called protein kinase G, or PKG. PKG, in turn, performs a crucial step. It phosphorylates, adds a phosphate group to various contractile proteins and ion channels within the smooth muscle cells. This phosphorylation leads, through a few steps, to a decrease in the concentration of intracellular calcium ions, Ca2+.
Lower calcium.
Right, and this reduction in intracellular calcium is the direct trigger for smooth muscle relaxation. Less calcium means less contraction.
Makes the muscle relax.
Exactly, and that relaxation allows the increased blood flow into the corpora cavernosa that facilitates erection.
Okay, so NO, Ni, SGC, cGMP, DCOG, lower calcium, relaxation, erection.
You got the cascade perfectly. Now, to your question about PDE5. Just as there's an enzyme to produce cGMP, SGC, there's also an enzyme whose job it is to break it down. That's phosphodiesterase type 5, or PDE5. This enzyme specifically degrades cyclic GMP, hydrolyzing it back to an inactive form.
The cleanup crew for cGMP.
Exactly. It essentially turns off the relax signal and allows the smooth muscle to contract again, ending the erection or preventing one from starting if NO signaling is weak. Therefore, the profound insight here, which led to the development of ED drugs, is that erectile dysfunction can be significantly improved by drugs that slow down this degradation of cGMP. How? By inhibiting PDE5.
Block the cleanup crew.
Block the cleanup crew. By blocking the off switch, these drugs allow the on signal cGMP to persist at higher levels and for longer, facilitating and maintaining an erection in response to stimulation.
Okay, now that we understand that intricate dance of chemical messengers and the role of PDE5, how do these incredibly well-known medications, the PDE5 inhibitors like sildenafil, actually step in to help based on that precise mechanism?
Right. With that foundation, the action of PDE5 inhibitors becomes very clear and quite elegant, really. The commonly available inhibitors of PDE5 that we use today are sildenafil, vardenafil, tadalafil, and avanafil. You probably recognize some of those names.
Yeah, Viagra, Cialis, Levitra. Those ones.
Oh, those are the brand names, exactly. All of these agents work by essentially competing for the same binding site on the PDE5 enzyme that cGMP would normally occupy to be broken down. By competitively inhibiting PDE5, these drugs prevent or slow down the rapid degradation of cGMP.
They get in the way of the breakdown.
They get in the way. This allows cGMP levels to remain higher and for a longer duration within the smooth muscle cells, provided there is stimulation producing NO and thus cGMP in the first place.
Ah, so you still need the initial stimulation. They don't cause it on their own.
Absolutely critical point. They don't initiate an erection spontaneously. They enhance the body's natural response to sexual stimulation by sustaining that crucial cGMP relax signal. It's also worth mentioning again that PDE5 inhibitors have that other significant therapeutic use beyond ED. They are also effectively used in treating pulmonary arterial hypertension. Same mechanism, relaxing smooth muscle, but this time in the lung arteries.
Right, different location, same basic action.
Exactly. In terms of their pharmacokinetics, how the body handles them, these drugs are generally well absorbed orally and are widely distributed throughout the body. They usually act fairly quickly, often within about 30 minutes to an hour after ingestion, though this varies slightly. While they all share the same core mechanism, they do differ somewhat in their affinities for PDE5, their time to onset of action, and crucially, their half-lives. This provides patients and clinicians with options.
Options for timing.
Right. Options to tailor treatment to individual needs and preferences for onset and duration. For example, tadalafil, Cialis, stands out with a significantly longer half-life of about 17.5 hours.
Much longer.
Much longer compared to sildenafil, Viagra, or vardenafil, Levitra, both of which have half-lives of around 4 hours. This longer half-life contributes to tadalafil's much longer duration of action, sometimes earning it that nickname, the weekend pill.
Ah, okay. Makes sense. How are they broken down?
These medications are primarily metabolized or broken down by the hepatic CYP3A4 enzyme system in the liver. That's a major pathway for many drugs. There are sometimes minor contributions from other enzymes like CYP2C9. Their metabolites are largely excreted via the feces in bile, with urinary excretion playing a secondary role.
Okay. And do they work well?
Clinically, yes. For most patients, they achieve satisfactory results, significantly improving erectile function. Starting dose recommendations vary for each drug, and it is always advised that patients begin at the lowest recommended dose, especially those over 65, to minimize potential side effects, titrating up if needed.
Start low, go slow. What about side effects?
Speaking of adverse effects, they are generally similar across the class due to their shared mechanism of PDE5 inhibition. But subtle differences exist based on their specificity, how much they might affect other PDE isoforms found elsewhere in the body. Common complaints include headache, flushing, dyspepsia, indigestion, nasal congestion, and dizziness. These are often related to vasodilation elsewhere. Back pain is also a known side effect, particularly associated with tadalafil, perhaps due to its longer duration.
Any vision issues? I've heard about that.
Yes, some patients using sildenafil or vardenafil in particular may experience transient blurred vision and sometimes a distinctive blue-green tinting of vision. This peculiar effect is due to some inhibition of retinal PDE6, a related enzyme involved in phototransduction, how light is converted into signals in the retina. Tadalafil is more selective for PDE5 over PDE6, so this is less common with it.
Interesting. Blue tint. What about drug interactions?
Yeah. Drug interactions are a critical consideration. Concomitant administration of potent CYP3A inducers, drugs that speed up the CYP3A4 enzyme, like the antibiotic rifampin or some anti-seizure meds or St. John's wort, will generally cause substantial decreases in the plasma levels of PDE5 inhibitors, meaning the ED drug might be cleared too quickly and be less effective.
Makes them weaker.
Right. Conversely, CYP3A inhibitors drugs that slow down the enzyme's activity, like many protease inhibitors used in HIV therapy, certain antifungals like ketoconazole, antibiotics like erythromycin, even grapefruit juice will inhibit the metabolism of PDE5 inhibitors. This prolongs their half-lives and elevates their blood levels, increasing the risk of side effects. Dose adjustments might be needed.
Makes them stronger, potentially too strong.
Could do, yes. And now for the absolutely critical, non-negotiable contraindication.
Okay, underscore. Bold flashing lights.
Absolutely. Consistent with their mechanism of potentiating cGMP signaling and causing vasodilation, PDE5 inhibitors dangerously potentiate the hypotensive or blood pressure lowering effects of organic nitrate vasodilators. These nitrates, like nitroglycerin, isosorbide dinitrate, isosorbide mononitrate, are commonly used for chest pain, angina, or certain heart conditions.
Nitrates. Got it. Big no-no.
Combining PDE5 inhibitors with any form of nitrate can produce dangerously low, potentially life-threatening blood pressures leading to fainting, shock, or worse. Therefore, this combination is strictly, absolutely contraindicated. Every patient must be thoroughly screened for nitrate use before even considering PDE5 inhibitor therapy.
Careful safety check.
Paramount. A patient's underlying cardiovascular status and concurrent use of any other hypotensive agents, like alpha blockers used for blood pressure or prostate issues, must also be very carefully considered and monitored before initiating PDE5 inhibitor therapy, as there can be additive blood pressure lowering effects. Finally, priapism, an erection lasting longer than 4 hours, is a serious, albeit rare, adverse effect induced by PDE5 inhibitors.
The 4-hour warning.
Exactly. It carries a significant risk of ischemic damage, lack of blood flow and oxygen to the cavernosal smooth muscle and tissues within the penis. It's a medical emergency and absolutely requires immediate medical attention to prevent potentially permanent damage and future erectile dysfunction.
Okay, good to know the risks alongside the benefits. Wow, what an absolutely incredible and comprehensive journey we've taken today. Seriously. From the very first moments of life to the intricate complexities of aging, and from the basic molecular building blocks to the cutting edge of pharmacological intervention, testosterone and its related compounds are truly at the heart of so much of human biology.
It really is quite staggering when you lay it all out.
We've gone from understanding exactly where these hormones come from and how they're meticulously produced to tracing their nuanced effects throughout our entire lives. We've seen how they shape our development, our physiology, and even our daily experience. And then we dived into the incredibly intricate ways medicine intervenes, whether it's to boost levels when they're deficient or to carefully rein them in when their activity is excessive. It truly is a testament to the stunning sophistication of our bodies and, the relentless pursuit of science to understand and harness these biological marvels. I feel like I've just completed a masterclass.
Aha! Well, if we connect this all back to the bigger picture, it really underscores how deeply interconnected our hormonal systems are with virtually every other system in the body. We've
Hmm.
seen how they influence everything from bone density and muscle strength to mood, fertility, red blood cell production, and even highly specific functions like those complex signaling pathways involved in erectile function.
Everything seems connected.
It really is. Understanding these pathways at this granular yet hopefully accessible level is not just academically fascinating, it's absolutely crucial for both accurately diagnosing and effectively treating a wide array of conditions. It really helps us appreciate the subtle differences between direct androgen receptor activation, those potent DHT-mediated effects, and even the surprising estrogen receptor-mediated actions that originate from what we typically think of as just a male hormone.
That multi-tool analogy again.
Exactly. And this profound interconnectedness and multifaceted action, well, it raises an important question for you, the listener, to perhaps mull over long after this deep dive concludes. What new insights might emerge as we continue to unlock the even more subtle interplay of hormones, not just in reproduction, but across all systems of the human body? We're still learning so much. And how might that deeper understanding continue to shape the future of medicine, fundamentally altering our approach to health, disease, and overall human well-being? What's the next frontier here?
Indeed. A great question to ponder. We hope this deep dive has not only given you a truly professional level understanding of this vital and fascinating topic, but has also left you not just informed, but genuinely inspired by the sheer complexity, elegance, and profound impact of human biology and pharmacology. Thank you so much for joining us on this deep dive.